human life receptor for how human life receptor for the invention of immunopathological consequences in Mukherjee malaria patients.
Thank you, Chair and from the audience. So today I will be speaking on the mechanism of immunopathogenesis in lymphatic filariasis and the possible target that can be used for developing drug against this neglected problem. So currently more than 40 countries are affected with lymphatic filariasis. It is a vector-borne parasitic disease, mainly transmitted by the mosquito. And in India, it is a big problem because India alone contributes more than 40% to the global disease burden of lymphatic filariasis. The immunology of this particular disease, parasitic disease, is very complex, but I am interested on the host-parasite interaction. That means after entry of the parasite inside the human body, this parasite interacts with a number of immune cells. Out of these, macrophages are the key player. So when the parasites are interact, I mean the parasitic antigens present on the surface of the parasite interacts with the macrophages. Macrophages change their phenotypes from M2 to M1. So the M1 are the classically activated macrophages that release the pro-inflammatory cytokines, that is TNF-alpha, IL-6 and IL-beta. And these three cytokines can activate different types of immune cells. As we know, they can trigger the activation, proliferation and differentiation of many immune cells present in different parts of the human body, including in the lymphatics. And as a result, the normal lymphatics turns into lymphedema and ultimately that forms elephantiasis. But as I said that it is a neglected disease because it is a problem of the underprivileged population. And another thing is that the people who are infected with lymphatic filariasis usually do not see that people are dying with this problem. But their life is becoming a curse. So till date, because when we started this work, there was no report that which particular factors from the parasite surface or which particular antigen is actually driving this phenotypic switching. So keeping this in our background, we started working on it to find out the possible antigen. So we collected the nocturnal blood because this is a nocturnally periodic parasite, only comes in the blood after 8 p.m. and it stays up to 3 a.m. in the night. So we collected the nocturnal blood and confirmed the parasite through thick blood smearing, and also we did molecular identification. And after that we collected the surface antigen lysate. And these surface lysate were added to the macrophage culture, the mouse leukocyte macrophage as well as the human THP1 derived macrophages. And before that, the LPS contamination was eliminated through polymyxin B and also checked by the LAL assay, which is a very common test. And we observed the level of the secretory level of the cytokines, pro-inflammatory cytokines in the spent culture media, and also the intercellular expression of the cytokines. And then we started purifying the antigen, which particular antigen is actually inducing the inflammation. First by ultrafiltration, and after that gel filtration, and finally using para-aminophosphatidylcholine by agarose binding chromatography, it's affinity chromatography. And through this series of chromatographic purification, I will tell you. So we have purified this particular protein. So how? So first we treated the macrophage with the crude lysate, and we have found there was upregulation of TNF-alpha release in the media. But we need to check whether this is a protein or not. So for that, we treated the protein with the proteases, that enzyme that degrades the trypsin, and also we heat inactivated the fraction. And in both the cases, there was no such inflammation. So the factor is a protein. And when we did the ultrafiltration, the fraction which is having a molecular weight of more than 50 kD, it was active. And when we resolved through active affinity chromatography, it appeared as a protein of around 70 kDa. So we sequenced this protein through multi-top mass spectroscopy, and we found that this is a protein that is comprising of 399 amino acid. And this protein, again we confirmed that whether this protein is actually doing the inflammation. So the answer was yes, because the 3 cytokines levels are upregulated. So this was identified through bioinformatics analysis through BLAST-P, and we have found that this is a homologue of the bestrophin protein, which is found in the free living nematode, that is C. elegans. And we generated antibody against this, and we found that this protein is localized on the surface of the parasite, and it contains 4 transmembrane helices. So, as this protein is actually expressing TNF alpha, IL-6 and IL-1 beta, so these 3 cytokines are the product of NF-kappa B. So NF-kappa B is the transcription factor. When it is phosphorylated, it becomes activated. So we check the activation status of the phospho-NF-kappa B through immunoblood and immunofluorescence. So we have found that yes, there was activation of the NF-kappa B. Now this, when we applied NF-kappa B translocation inhibitor, we have found that there was no such expression of the cytokines. So primarily it was rooted through NF-kappa B. As you can see, the canonical markers like phospho-IKB alpha, IKB alpha, NEMO, and phospho-NF-kappa B, all were upregulated. Now the NF-kappa B is the transcription factor that present in the cytosol, and when it gets the signal from the cell surface toll-like receptor, majorly the toll-like receptor 4, because in the earlier literature I found that there is a role of TLR4 in the Bankov's and familial immunopathology. So we check the TLR4 expression. So when TLR4 is activated, its surface level is upregulated. So we have seen that the TLR4 upregulation was there, but when we knock down the TLR4 with siRNA, we have found no such inflammation. That means primarily this pathway is dependent on TLR4 and NF-kappa B axis. So the next and the very important question was that whether this particular protein, we termed is an MFP, that is microfilarial protein, which is present on the surface of the parasite. So whether it acts as a ligand for TLR4 or not. So what we did, we labeled this protein with FITC, and we applied this protein in the cell culture media in the macrophages, and at the same time we did the antibody blockage in the macrophages. That means we treated the macrophage with anti-TLR4 antibody, and we have found that there was no such binding. But in the MFP data set, the green signals are saying that this protein is binding to the cell surface TLR4, and this was confirmed through co-immunoprecipitation. And finally, we did the biophysical interaction between these 2 particular protein. So this protein-protein interaction was checked through surface plasmon resonance. So here you can see that there was a dose-dependent binding of this particular protein to the TLR4. And finally, we did the bioinformatics analysis. We've seen that this particular protein is binding to the extracellular domain of toll-like receptor 4. So that means this protein happens to be a novel ligand for TLR4, that binding to the extracellular domain of TLR4, and it is causing the NF-kappa B activation and release of the cytokines. But the story is not over, because this parasite, the Ujiria Bankovki, the vector, in case of malaria, the parasites are directly discharged into the bloodstream, but in case of filaria it is not. The parasite first deposited on the skin, and then the parasite started migrating through the skin barrier. So beneath that skin we are having dendritic cells. So the question was that whether this protein has any role in dendritic cell activation, because the immunopathology is, in filaria itself, is a coordinated mechanism of activation of different immune cells. So what we did, we took the immature macrophages that were generated from monocyte, and we treated the immature monocyte-derived dendritic cells with this protein, and we have found that this protein can induce both maturation and activation, as there was upregulation of CD40, CD86 and HLA-DR on the surface of dendritic cells. But and it was also confirmed with these cytokines. But interestingly, when we added TLR4 pathway inhibitor, that is CLI-095, we didn't see any kind of activation. That means this process is it is TLR4 dependent. So for same. Now in this case also we have found, since we assume that this could be a ligand for TLR4. Here also we have seen through flow cytometry and also that immunofluorescence and co-immunoprecipitation, this protein is also binding to dendritic cell TLR4. Now when the dendritic cell gets a signal from the antigen, or it is educated by the antigen, it activates the T cell subsets. So herein we have seen that this particular antigen when it is processed by the dendritic cell, it causes the expansion of Th1 and T17 cells, and it was also supported by the cytokines. So this protein not only it activates the macrophages, but it is also having a role in the dendritic cell activation as well as maturation. So can we target this particular protein? Because our aim was to develop a drug for that. And we are having so many chemistry collaborators in India. So these two particular biomoieties, so they are actually working with these two type of scaffold, as quinolone and sulfonamides. They are well-known bioactive compounds. So I told them that whether we can develop by fusing these two type of scaffold in the laboratory, so which can be effective, because these quinolones, sulfonamides, these sulfonamides, their absorption rate is very low, but they are very active. At the same time, quinolones their absorption rate is very high. So they prepared a group of molecule, out of which we screened this molecule with our German collaborator in the University of Bonn with Professor Kenneth Farr, and we reported this particular compound for its supreme antiviral activity. This already is active. But the problem with this particular compound is that it is doing very fantastic activity in case of in vitro system, but its in vitro absorption rate is very poor. So we started modifying this particular compound, and we added several groups. But this particular agent 3, when we added this dihydroxyphenyl, we found that this is a very active one in terms of killing the parasite as well as reducing the inflammation. Because when we treated the cells with LPS and after that we post-treated with this particular compound, we have seen there was reduction of the inflammatory parameters. So, and this particular molecule is binding at the same place where the phenyl protein MAP binds. That means the particular, it is a quinolone fused cyclic sulfonamide that can be used as a potential antiviral agent. So, but apart from this, since the protein it is having a very high degree of inflammatory activity, so can we develop vaccine against this particular protein? So keeping in that view, we actually developed the anti-sera by treating the rat or mice. We taken the serum and we treated the parasite. We have seen that it can reduce the worm load. So, and also important thing is that when we treated the Uchera Bangkokite parasite isolated from the human blood, we treated the parasite with this particular serum, and we have seen that this antibody treated parasites have failed to induce any kind of inflammatory signal. So this is a very preliminary work. That means considering the pro-inflammatory nature of the protein that I have shown to you, it can be a vaccine target, because this is a very preliminary data. When we treated the mice or rat with this particular protein, we have seen that it can protect the parasite, protect the animal from the parasitemia. And right now we are actually working on this to find out the B cell and T cell epitope to actually to ligate those epitopes to form a multi-epitope vaccine. So these are some of our publications with our collaborators and from our laboratory. These are my collaborators and funding agencies, and this is my laboratory, and my student Ritik is there in the audience. So thank you all for your patience.
Thank you very much for the nice presentation. We have room for 2 questions. I will start with the first one. If we suppose that total receptor 4 activation would exacerbate lymphatic filariasis, leading to lymphedema or lymphangiasis, how can you explain the fact that 80 to 90% of the infected individual have low pathology or completely asymptomatic?
Yes, because as you know, you're also working with lymphatic filariasis. The immunology is very complex, and it differs from demographic area to area. And you see actually the initiation stage of the infection and the cure stage, these 2 particular stages that are actually showing the higher inflammatory status. But in the middle stage, the chronic infection, you see there was more anti-inflammatory nature. So we need to stop the disease either in the infection stage, that means during the initiation stage of the infection. When the infective stage, that is the L3 larvae, enters into our body, in that point, you see the patients actually suffer from fever, sudden fever in the midnight. So in that portion part, we need to recognize that. Otherwise, if that parasite matures to adult, you know that there is no medication right now.
Thank you. Another question? Okay.
Great talk, thank you.
We need two more questions.
So it wasn't surprising actually with the dendritic cell that just that it's going to be signaling to toll-like receptor 4 as well. But what I was wondering was, you use antibodies to show that it's toll-like receptor 4 dependent. Did you try to knock down TLR4 on these cells and try to repeat the experiment? Because the antibodies could be doing something different. Yes. Did you do that experiment?
Yes, we did some with some TLR4 pathway inhibitors. Okay, but we didn't do that siRNA because our aim was to find out the role of this particular protein in DC activation. Okay, because yes.
Yeah, I was wondering because I think you should have gone back to try to see when you knock down the TLR4 expression of the cells, because there may be other toll-like receptors that they may be working through, or even other receptors you don't know. So you could have knocked down. And then look to see that when you now incubate those cells with this molecule, now you are bullying the production of those pro-inflammatory cytokines.
Yes.
That would have been a very good definitive experiment.
Thank you.
Well, thank you. That was very interesting. I was just wondering, could you commonly use anti-inflammatory drugs that suppress NF reactivation to kind of prevent the inflammation that causes the disease?
Actually, you see that in case of highly inflammation, TLR4 plays a major role. So, if you take antiviral drug, if you kill the parasite, there is a chance, or not the chance, actually the fact is that the parasite will release the LPS, because this particular parasite, the biology is very complex. The parasite contains one bacterium, that is Wolbachia, and when the Wolbachia release in the blood, the patient, and this particular gram-negative bacterium, it contains LPS. So from the dead parasite, LPS is released in the bloodstream, and that can cause septic shock. So this is a problem. I don't know, but nobody has still, I think in literature, nobody has done the therapy with anti-inflammatory drug. Or if you treat with anti-inflammatory agent, there is a chance that parasite can migrate towards deeper circulation and it can copulate and it can produce many parasites.
And do you know if that would happen with your drug as well?
So yeah, that can be possible, but at the same time, this drug is killing the parasite also. There is a beneficial effect of this drug.
Thank you. We will